population. So they wanted the best of both worlds, uniqueness and similarity simultaneously. Other paleontologists initially looked at the fossil record, not even looking at the DNA record, and said, I got a Chinese and a South African fossil here that are very, very old, and yet they look almost indistinguishable, like they could practically be cousins. So from this single recent replacement model was proposed, in which it was, yes, Homo erectus did go into all the other areas of the old world. It gave rise to various populations, including the Neanderthals, but it was only one population of Homo erectus through the Archaics that became anatomically modern, and when they migrated, they completely replaced the existing populations. So this was initially proposed based on the fossil record. In the late 1980s, though, a new series of studies first came out, and one of the most seminal of them was put forth by Rebecca Kahn, then a graduate student at UC Berkeley under Alan Wilson's tutelage. And she examined mitochondrial DNA from people from all over the world. And one of the things you can do with any type of DNA, but particularly mitochondrial DNA, is you can trace the diversity that exists in the present back until it has a common ancestor. And this is any stretch of DNA, any gene, the different alleles, the different versions of that gene will share a common ancestor. It could be a couple thousand years ago, it could be millions of years ago, it could be billions of years ago. This common ancestor, this coalescent, it could be a bacteria. These others looked at mitochondrial DNA. Mitochondrial DNA is purely maternally inherited. It's actually passed on in the egg. None of the mitochondria in the sperm, or effectively none in the sperm, get passed on. So it is a so-called her story. It is the story of our maternal origins. And when she sequenced 146 people from around the world, she found 135 different kinds of mitochondrial DNA, so incredibly diverse. If I would sequence everybody in this room's beta globin gene, I'd come up with maybe 4 alleles. Mitochondria, I would come up with many, many, many more alleles. When she inferred an evolutionary tree based on these mitochondrial sequences, she found that the deepest roots, all of the individuals at the very deepest roots, were of sub-Saharan African ancestry. And then Africans and people from elsewhere in the world made up the rest of the tree. And to this suggested to her that there was an African origin and that all the people on the planet derived from Africa. Now, this alone wouldn't be shocking because all of the Australopithecines, all of the earlier hominin fossils are African. What was shocking, though, was she knew the mutation rate of the mitochondria, and by counting up the number of mutations in this tree, she inferred that this common ancestress of all modern people was less than 200,000 years old. And the press, I hope there's no journalism majors here, or if there are, you need to learn science. The press picked up upon this and dubbed this single female ancestress of all of us mitochondrial Eve. And this is where the mitochondrial Eve hypothesis gets its name. But it's not that there was a single woman. She had cousins and daughters and sisters and aunts and all sorts of other people living in her population. It's just hers was the single one that actually survived to this day. It's like when you're at a family reunion and you get a picture of all of the cousins together. The reason you're cousins, you share at least one grandparent. But you don't share all of your grandparents, and so that's why you're cousins. So there were some serious flaws with her study, both in the samples that she collected, may not have been perfectly representative of all of humanity, because it was basically people from all over the world who lived in the San Francisco Bay Area. So they weren't perfect sample. She used some rather weak analytical techniques. They were the state of the art at the time, but then they were weak. And so some people criticized her study, but since her study was carried out, a whole series of studies have been carried out that demonstrate both that there's more diversity within Africa than the entire rest of the world. The rest of the world's diversity is just a subset of that found within Africa. And if you build proper trees out of it, you find that the deepest roots of the trees indeed are in Africa. So overcoming all of the methodological shortcomings of the initial studies of Becky Kahn and Linda Vigilant and others, and they weren't, again, wrong at the time. They just, it was the techniques that were available. She didn't even have PCR when she started that. She was actually doing it from restriction mapping of whole genome, mitochondrial genomes. And the analytical techniques to infer these trees were rather difficult. Nowadays, new techniques, which I won't go into in detail, allow us to infer these techniques with much larger data sets and much better. And this now is more or less the indisputed story of human mitochondrial DNA. If you look around the world, there are basically three main groups of mitochondrial DNA, and these are represented by these blue spheres here, and there's some subgroups within them. But the entire rest of the world, all three of these groups, these are called mitochondrial haplogroups. All of the individuals within those spheres are more closely related to each other than to any other sphere here. All six of these blue spheres are found within sub-Saharan Africa. All of Europe, Asia, and South Asia are found derived from a single group. So I like to say if a biologist from Alpha Centauri landed on Earth and they had special goggles that only allowed them to see mitochondrial DNA, and they were taxonomists and wanted to divide humanity up into groups, they would either have 3 or 6 groups. One of those groups lived everywhere on the planet, and 5 of those groups only lived in Africa. And so the mitochondrial story is somewhere around 200,000 years ago. A population of archaic hominins got lucky. They spread throughout all of Africa, dividing up into the other major haplogroups. And around 55 to 60,000 years ago, a single relatively small population of those people migrated out of Africa and gave rise to the entire rest of humanity. And so now this no longer is a contentious story. It was as it was being developed, and the problems of the techniques were elucidated and all that. But now this story is kind of set. The problem is, it's a single story. It would be like me asking you about your ancestry just by asking about your dad's great-grandfather. You have many different great-grandparents who come probably from all over the world. If I just ask you, well, actually your mom's grandmother would be the better analogy. That would only be one part of your story. I would be German if that was the story. I wouldn't have my French and Irish and British and all those other things in my story if I only looked at my maternal ancestry. Because the different components of our ancestry, the different components of our genome, in fact, each independent stretch of DNA that under Mendel's law of independent assortment doesn't link with others, has its own unique evolutionary history. So this is what we refer to as the gene tree versus the species tree. It'd be like saying I am whatever my maternal grandmother was, ignoring the other 7 great-grandparents. So it's a clean story, but it's only part of the story. So another part of the story can be gleaned just in men, and that is through our Y chromosomes. So the non-recombining portion of the Y chromosome, all of the dudes in here have it, and we got it from our father and they from their father. So just like the mitochondria, even though I have mitochondria that I inherited from my mom, because I had 2 sons and no daughters, my mom's mitochondria is extinct with me. On the other hand, I am passing on several copies of the Y chromosome into my offspring, so we can figure out the paternal history. Now, the Y chromosome is very interesting, because at one time it was thought to have almost no diversity. It was thought to only carry 1 or 2 important genes and have very, very little variability. As new technologies were discovered, we were able to find out a tremendous amount of variation in the Y chromosome. And when they did that, it turned out that all humans on the planet share a relatively recent male ancestry that left Africa sometime between initially estimated 35 to 89,000 years ago. But this is spot on with the mitochondria story. So if you create and infer trees, you see that basically all of those ancient Y chromosomes found in Indonesia and China and Europe and elsewhere are not on the planet today. And only 50,000-year-old or so Y chromosomes are found, and they all trace back to Africa. So the initial hypothesis that, well, maybe that if the mitochondrial story was true, maybe females were spreading all over the world, but they were still hybridizing with males all over the world. This dispels that part of the story. This tells us that both our male and female lineages go back to Africa. So, basically Eve, African Eve, could have known African Adam back there in Africa in the last between 100 and 200,000 years ago. Over the last 20 years, we've dissected other parts of the genome, and they all suggest a common and recent African origin for all non-African human populations. So region after region of the genome suggests this. Now, this is really important because, as I had pointed out, and in fact I stole this from Willy Hennig, an East German entomologist from the 1950s, each different component of our genome, as I said, has its own unique history. And if all of those histories coalesce together into a similar one, that probably tells us we now know the actual species-level phylogeny and not just an individual gene or allelic phylogeny. So we can solve the so-called gene tree, species tree issue by looking at many, many, many independent genes. And they're not all going to go perfectly, but the vast majority of them coming together will tell us that. So using dozens of different techniques of dissecting the human genome, we keep finding a recent African origin spread across all 23 different chromatids in the genome. And no matter how we do it, we keep coming up with the same story. We can actually look at hitchhikers. So in many of our guts, we carry a bacteria called Helicobacter pylori. And if we reconstruct and you only inherit this, you get infected from this as an infant from your mother. You can be married to somebody for 50 years and never pass on your gut bacteria to them. But your son or daughter will have the mother's Helicobacter types. When they reconstruct the evolutionary history of Helicobacter, it is the same as human history. Deep roots in Africa, recent derivations of Europeans and East Asians from Africa. In your mouth, you carry a bacteria called Streptococcus mutans. Again, no matter how much you make out with somebody, you're not going to pass this on easily. You are going to have your mother's Streptococcus mutans. These are the bacteria that actually cause cavities. And if we reconstruct their history, we see deep African roots, and non-Africans have basically recently derived African subtypes. So this really lays out a clear story that we don't go anciently back 1.8 million years ago when Homo erectus first reached China, Europe, Indonesia, and elsewhere in the Old World, and that the vast bulk of our genome is only 200,000 years old or less, and those of us outside of Africa even more recent. And it explains some patterns of human variation. If you want to do a study to test how variable are human populations, it'll cost you just a few dollars. Because what you need is a globe and a tape measure. If you measure the actual geographic distance that people would have to migrate to get to different regions of the world, and then you steal somebody else's data on their genetic variation, there is a direct linear correlation. Biologists kill for an R of 0.9. I mean, that never happens. And yet, the only way you could explain this is if there was a recent origin and all of the people all over the world migrated out of there, and due to genetic drift and the founder effect, slowly lost the variation as their populations expanded outside of Africa. So we have a very, very strong picture of what it looks like. It might be subtly more interesting. There might have been 2 migrations out of Africa somewhere between 50 and 60,000 years ago, a so-called southern route and a northern route. But they both came from more or less the same place, almost the very same people. So one of the reasons all of the genes of non-Africans look so similar to each other is they're probably derived from the very same African population. Now, what do the fossils tell us? Well, the world's oldest anatomically modern human fossils are found in Ethiopia, South Africa, 110,000 years ago, 120,000 years ago. In Northeast Africa, 95,000 years ago. And surprisingly, in the Middle East, around 90 to 100,000 years ago. But at that time, the Middle East was basically a faunal province of Africa. So all of the animals living in the Middle East looked just like the other animals living inside of Africa. And the so-called famous Cro-Magnon people of Western Europe, latecomers, dead end, 35,000 years ago. So, 30 years ago, I would have been giving this lecture and say the earliest anatomically modern humans came from Europe. Now we know that the Europeans and the Far East Asians are the very last anatomically modern people to appear in those regions. But, at least in Europe, and a good chunk of Asia, at least up and through Siberia, there was another group of people, the Neanderthals. This is actually the oldest Neanderthal specimen ever discovered from 1848 in Gibraltar, though they didn't know what it was at the time. By 1856, they had discovered the original specimen. This is what Neanderthals are named for. This is the Neander Valley specimen from Feldhofer Cave in the Neander Valley. Neanderthal in German means Neander and Tal is valley. And this is the type specimen for Neanderthals. Now pay particularly attention to the humerus because it gets quite interesting. This is another specimen from France of Neanderthals. And the Neanderthal fossils, the classic Neanderthal fossils, were existing. They were found in Europe between 200,000 years ago and about 30,000 years ago. Their potential predecessors might go back to 500,000 years. That's a little bit more contentious. Tying fossil lineages directly to each other is actually rather difficult. But there are things 400 and 500,000 years ago in Spain that look kind of Neanderthal-like and not quite anatomically modern-like ourselves. But whatever the case may be, Neanderthals existed in Europe at the same time that modern humans lived in Africa, and they overlapped for thousands of years, depending on when Neanderthals first got into the Middle East and when modern humans first got into Europe. But anywhere from 10 to 50,000 years ago, these two groups of distinctively looking people would have overlapped in time and space. So anybody in evolutionary psychology could easily answer this question. You know, if they ran into each other, yes, they would have been likely to have had sex. What was the result of that sex is the more interesting question. Are there hybrids? So some paleontologists claim, for instance, this 28,000-year-old or 26,500-year-old jaw from Portugal up in the mountains is actually evidence of a Neanderthal-human hybrid. Highly contentious. Many people argue against it. It's a 5-year-old. Child skeletons don't tell you as much as adult skeletons. And there's another dozen or so jaws and teeth and skulls that some people claim are hybrids, and others said, no, that falls within the realm of all the variation that we see around us anyways. So, this question began to get more interesting in 1997. In the summer of 1997, a group of researchers in Pennsylvania, Munich actually got permission to take a large chunk out of the original Neanderthals' femora, and from that, using the technique of the polymerase chain reaction, PCR, what we use in forensic contexts and so on, they were able to piece together and reconstruct a sequence of DNA that basically did not overlap with modern humans, fell out of the range of chimpanzees, and they claimed, they inferred, was a legitimate Neanderthal sequence from that specimen. Now, this was really, really contentious at the time. Was this contamination? Was this an artifact due to the degradation of ancient DNA? All these things we know happen. So was this artifactual? And one guy even argued, well, look, there's overlap. Again, most biologists would be happy if they had this kind of division. And it's only one specimen, as if paleontologists have never claimed something based on a single skull. So, just about 9 months later, another colleague of mine was able to sequence another Neanderthal specimen from the Caucasus Mountains in southern Russia. And lo and behold, his sequence joined with the German sequence and fell outside the range of all modern sequences. So it wasn't that the Neanderthals linked up with Europeans, which the multi-regional versions of the model would say Neanderthals and modern Europeans would be related to each other, but not to other populations. It fell outside the range of all. And when you put a date on this tree, this was over 500,000 years ago. So before the appearance of Neanderthals and well after the appearance of modern humans, so that these guys would be just as much a side branch as the robust Australopithecines and other lineages that clearly aren't our relatives. A couple months later, a third Neanderthal sequence was sequenced, and then a fourth and a fifth, and on and on. And we now are up to, probably, it changes every month or two, but there's at least 24 different Neanderthal mitochondrial sequences that have been collected. One of the reasons they had to do it with mitochondrial DNA is mitochondria are special because every cell has hundreds to literally thousands of copies of mitochondrial DNA, but only one copy of the DNA in your nucleus. So if you're going to be working with 40,000-year-old degraded DNA, you want to have a 1,000-fold chance of finding it than finding that one Y chromosome. So for the first decade of archaic genetics, we could only work with mitochondrial DNA. Now, a early anatomically modern human was also sequenced at this time, a so-called Cro-Magnon. But there was a huge hue and cry about, well, you can't tell if this was a contaminant, because there's always contamination from the excavator, the guy at the museum, your lab tech, or whatever. I used to like to say that until we train chimpanzees to actually excavate and carry out lab work, we're never going to be able to truly distinguish contaminating DNA from the modern period to that of our sample. So this study was dismissed because they said, well, you can't rule out contamination, which at the time was a fair criticism. But a neat partial workaround by the same guys who were criticizing that and who had discovered the initial Neanderthal DNA, they came up with, well, not a perfect experiment, a quite ingenious experiment. Because there were differences in the DNA, mitochondrial DNA of Neanderthals and modern humans, they developed an assay using PCR, using special primers. They had a set of primers that would work on any hominin or hominoid sample, but they also had a special set of primers that would only amplify Neanderthal DNA. And so this is kind of an absence of evidence argument, but they brought in cave bears as well. And by sequencing these cave bears, they demonstrated that the caves in which these bones were found in had good enough preservation conditions for cave bear DNA of exactly the same age as the Neanderthal and early modern human specimens. So high likelihood that those specimens would have DNA. And the Neanderthal remains, and the ones that morphologically looked like Neanderthals, all yielded Neanderthal sequences, but none of the anatomically modern samples, which did have DNA in them, yielded Neanderthal DNA. So from this, they concluded that if you go back 30,000 years ago, when these guys could have hybridized, there was no cross-connection between the Neanderthal and modern human mitochondrial genomes. So not a perfect experiment, but a pretty ingenious one that suggested that there was not Neanderthal DNA in even the very earliest moderns who would have been hybridizing with them at the time if that theory was correct. And so again, this is mitochondrial DNA. This is the maternal story. But because they got so good at amplifying Neanderthal mitochondrial DNA with Neanderthal-specific markers, they began to apply it to all sorts of samples that the bones were just too crappy and scrappy to figure out what they were. And they actually discovered Neanderthals all the way into Siberia. So when I was a kid, Neanderthals only existed in Europe and the Middle East and, far or in Central Asia, but that was the range of the Neanderthals. We now know Neanderthals lived all the way into Siberia, quite a distance. So one of my grad students and I, summarizing all of that data to date, made a bold statement and prediction. We're wrong. But at the time, we made this bold statement and prediction, taking all of the data that had been known today, all of the sequences that had been known today, we demonstrated that basically at the mitochondrial level from the female lineage, there was complete and total African replacement of the mitochondrial DNA. There wasn't complete multi-regional evolution. there wasn't even a little bit of hybridization with the Neanderthals based on the original mitochondrial data. So no Neanderthal females passed their DNA on to the future. Now, some really cool things began to occur. We started to get a little bit better at extracting the DNA from these samples to the point we could begin to get scrappy DNA from the nucleus. And one of the first genes sequenced was the melanocortin-1 receptor. And is what's really neat is some Neanderthals had red hair and light skin and freckles. They had a mutation in the MC1R gene that yields this in modern humans. So you'd say, well, there's clearly evidence of hybridization. Well, it turns out it is a completely different independent mutation that knocks out the gene in the same way that yields this result. So even the gene that everybody said, redheads must be Neanderthals or have Neanderthal ancestry, even that turned out to be parallel or convergent evolution, which was a really, really cool story. So, lots of people modeled this, lots of people looked at this data, and these two names will come up in a few more minutes. They looked at this and they said, based on the probabilities of the number of sequences gathered, what's known about human mitochondrial DNA, basically there is no Neanderthal DNA, well, at least mitochondrial DNA, in the current human genome. And that still holds true. There is no, in the tens of thousands of modern humans who have been sequenced to date, and in the 24 or 30 Neanderthals who have been sequenced to date, there is no overlap in our maternal, purely maternal history, in our mitochondrial history. But all of this was torn asunder when new technology kicked in. And so using next generation or second generation sequencing techniques, which instead of having to amplify the DNA, can actually work with tiny, short, little damaged fragments of DNA, in 2010, a draft sequence of the entire Neanderthal genome was sequenced from these three wonderful specimens. I hand these to any paleontologist, and they're like, rib, maybe, possibly tibia, I'm not sure. But they had lots of DNA in them. They carried out all sorts of neat experiments, and they found something incredibly shocking. They found variants of Neanderthal alleles were found in the French, Chinese, and Papua New Guineans, but not in the Yoruba and San. So they found Neanderthal alleles, a very tiny percentage, 1 to 4% of the alleles in the Eurasians were shared with Neanderthals, but not with the Africans. And so from this, they came up with several scenarios. Either there is really deep ancestral complex population division in Africa for hundreds of thousands of years, or their theory was that there was a hybridization event after moderns left Africa, but before they split off into Asia and into Europe. So they're predicting it happened right there in the Middle East. And because of this kind of research now, my poor paleontological colleagues, at every dig they go to, have to dress up in full moon suits, because the most likely point of contaminating a sample is upon excavation. Once you sort of dig it out of the ground and expose it, that's the worst possible time. So now there's lots of people excavating dressed up like this. And they've now testing dozens and dozens of samples. We now can actually look at individual genes, just like that skin color and hair color gene. We can look at a whole bunch of other genes. We're beginning to look at genes of the immune system and all sorts of other things to begin to tease apart what Neanderthals were like. And indeed, is this a false signature of hybridization? or is it indeed real? So the average person in this room who's of Eurasian ancestry has, on average, about 2.5% of your genome is shared with that of Neanderthals. So that might sound like a whole lot, but in a moment I'm going to show you that it's actually not much. I'm going to skip over this, because those same two guys I had mentioned earlier have also run simulations on this, and through a process that I like to call surfing the wave, but it's more like the serial founder effect. If you can imagine an existing population, say in Europe, and a new expanding population coming out of Africa, if they were to hybridize, where will they hybridize? It's going to be right at the contact boundary. Now let's say they pick up, they hybridize, and a few alleles go back and forth between them. But this modern population is continuously expanding and pushing outward. Maybe we could have one more child per female, or maybe our technology was slightly better. Maybe we carried diseases that the Neanderthals weren't ready for. But every time we meet and pick up a gene, but then our population expands, it's like the founder effect. It's like just a very few individuals starting a whole new population. So by picking up just a few times these Neanderthal alleles into the expanding modern population, we carried them forth and amplified them. And so they've estimated it could be 200 to 800 mating events over 10,000 years across the entire range of connection with Neanderthals and modern humans could yield up to 2.5%. This isn't 2.5% of all the individuals were mating with each other and having offspring. This could be every few generations, one of these hybrids survived and passed on genes to the next generation. But because they were at the front, the wave, if you will, of this expanding population, they surfed it till that tsunami of anatomically moderns basically overwhelmed the existing Neanderthals in Europe. So now, while I was wrong about there being no introgression at all, it's still not the multi-regional model of plenty of introgression. And the question is, where did it happen? Did it happen right there in the Middle East at that moment, that very few years they were in the Middle East before they went to Europe and before they went to Asia? What if there was a South Arabian route and a North Sinai route? They would have been different populations. So my grad students and I have been looking at this, and one of our major criticisms of that initial hypothesis about the hybridization is here's their sample. 2 Africans, French, Chinese, Papua New Guineans, 1 each, and 3 Neanderthal scraps put together into a composite genome. This is what they deduced that from. We said, well, just from that scrappy data, you don't know if it happened in the Middle East, if it happened in North Africa, and without sampling more of Africa, we don't know how many Neanderthals alleles are there in Africa. Just this afternoon at lunch, I downloaded a new article, I didn't have time to incorporate it, that was suggesting they found some Neanderthal alleles in modern Kenyans. So, and that's exactly what we predict in this paper, is that if you sample North and East Africa, those very same Neanderthal alleles you're saying are all over the world are probably also going to be found deeper in Africa. So we don't know the story of this yet, but it's now interesting. We know there was hybridization. Now, really recently, another study was done looking at a whole bunch of African individuals and parsing their genome and carrying out a complex analysis and a model of how the variation in several hundred African genomes came to be. And they found a signal of what they said. Well, most of them look about 200,000 years old at the most, but there's this tiny percentage that's even older. So the geneticists are now predicting another hybridization event with another group of hominins that we don't have a single fossil for yet in Africa. Because after all, hominins have been in Africa for 6 million years. Many populations, Erectus probably, East African versus West African versus South African. Erectus would have been quite different from each other. The archaic populations that they gave rise to. So there's probably hidden in the fossil record of Africa, or maybe not even fossilized because it's in the rainforest of Central or West Africa, there's probably the equivalent of a Neanderthal-like species of archaics that may have hybridized a couple percent, 2 or 3%, with sub-Saharan Africans. So, this sounds like crazy, and I would have said, yeah, well, show me the evidence, because it's just a model of how the variation arose. But then, just last year, 2 years ago, they discovered from a pinky, a fragment of a pinky and a single molar that doesn't look like anything else, in Siberia, they found a hominin, and when they sequenced its mitochondrial DNA, it's older than even the Neanderthal human mitochondrial DNA. And so now it's like, whoa, there's another species that left Africa, but it's all we have is this pinky and molar, and its DNA falls outside the range of even humans and Neanderthals. Well, they went on, and these are the Denisovans. So, similarly to naming the Neanderthals after the Neander Valley of Germany, this is the cave of Denisova in Siberia. So, for the moment, we're calling them Denisovans, like we say Neanderthals. And here we see the Denisovan cave. This is that Neanderthal cave I told you about. They're real close to each other. And the same group that did the Neanderthal work went in. Here's the tooth. Is all they have they have a pinky, a part of a pinky, some artifacts, and one molar tooth. But using these new technologies, they have now sequenced the complete genome from this scrap of pinky. So it was a youngster, because it was tiny. It was female. And it's distantly related to the Neanderthal, but more closely related to Neanderthals than to us. So what we refer to as the sister group of Neanderthals. But if you go down to Papua New Guinea, you find a couple percent of the Papua New Guineans' DNA matches that of these Denisovans, which is just really freaky. How do you go from Siberia to Papua New Guinea? Well, part of it's sampling. When they began to sample more people, they found it in Indonesia and Thailand and Cambodia, these alleles, but they don't find it in India or East China. So there seems to have been a corridor down all the way to Australia. So the Australian Aborigines have this archaic population's DNA, just a few percent. They also have Neanderthal DNA, a few percent of that, but it's now gotten quite interesting. But the Denisovans, so the Neanderthals hybridized with the ancestors of all Eurasians, the Denisovans only basically of Peninsular and Oceania. Peninsular South Asia and Oceania. So they weren't as widespread as the Neanderthals. But it's getting really interesting. But what about that mitochondrial DNA that's even more ancient? My pet theory, now, like 3 of my pet theories have already been shot down, but my new pet theory is that there was another archaic that had that Denisovan mitochondria running around East Asia. That population now is extinct, But before they went extinct, like the Neanderthals and Eurasians, they hybridized mitochondria. If it gets through the species boundary, it's really easy to spread throughout a species because of its maternal inheritance. So maybe so we now have running around the planet, 50,000 years ago, Denisovans, Neanderthals, and Denisovans. Maybe their archaic ancestor was still alive, but we don't have any other evidence of it except its surviving mitochondrial genome. We don't know for sure, but nevertheless, it looks like it, might be even a fourth species. So when people talk about, the missing links in the human evolutionary tree, it's more like a mesh or a bush. We now got too many of these guys running around, and it's really difficult to say exactly who was it, because there was some introgression and gene flow going on back then. But if we look at, again, these models, it was a very, very tiny amount of gene flow going on here. So we're talking, hundreds of mating events, not tens of thousands of mating events, over, 15,000 kilometers, over 10,000 years. This wasn't just continuous interbreeding. And so, in one of my recent papers, I've tried to sum up this mess graphically, and probably haven't done a very good job of it. So we have Homo erectus first appearing in Africa, spreading throughout Europe, Asia, and Africa. We actually don't know what's really happening in this period in Asia. We just have no fossils at all. But we have some in Europe that may have given rise to these archaic populations. One of these archaic populations probably gave rise to the Neanderthals. When moderns first appeared 200,000 years ago in Africa and slowly spread out, they ultimately run into the Neanderthals and their distant cousins, the Denisovans, swap a few genes, and then just go extinct. And now it's all modern humans. And my fourth utterly hypothetical hypothesis is the little hobbit in Flores Island in Indonesia. I don't think it's a microcephalic modern human. I think it's a pygmy, late-surviving Homo erectus. And so that's what this little stream is. But this is purely hypothetical, because we have no clue what these guys are. And because it's so warm and wet there in Indonesia, every time they try to get DNA out of these specimens, they've just not been able to. The Neanderthal and Denisovan, we're talking Northern Europe and Siberia. It's like keeping it in the icebox. Keeping it in Indonesia, it's like keeping it in, a sauna. And DNA is just not going to survive under those conditions. So, it's now really interesting, though, if you think back between 50 and 200 or more thousand years ago. There were 4 or 5 different species of, do you want to call them human, proto-human, some not? Again, where are we going to divide the line between us and others? But nevertheless, we share portions of our genomes with some of these guys, some of us more than others, some of us maybe not at all, and I am sure with these new technologies and as we sequence more and more genomes, we're going to find a far more complex story, and our story is going to get much, much more interesting. So, I'm sure you don't have any questions, but I'm more than willing to entertain some. I can quickly show you another theory of mine that's wrong. Let me jump ahead here. We'll skip the Australian genome, which they got out of a lock of hair. Kind of interesting. So, I had predicted in my Chumanzi paper. Chimps, gorillas, and orangs have 48 chromatids. We have 46. Most people assumed that fusion occurred right after we split from the chimp lineage. I hypothesized in the paper that maybe that fusion event that created human chromosome 2 only happened 200,000 years ago in one particular population of archaic hominins in Africa, and that gave rise to modern humans. And so that spread around, and when we left Africa and ran into the Neanderthals in Europe, at the time I didn't think we hybridized at all. We had incompatible chromosomal complements, and that could explain why we didn't hybridize with them. Well, with the new Denisovan genome, it looks like Denisovans and equally likely Neanderthals probably had the same chromosomal complement that we did. So it was a fun theory, but it looks like it's not going to hold based on these new genomes. So now I'll take questions. Yes? Yes? Well, so in 1859, when he wrote The Origin of Species, there wasn't a single reference or mention of humans. Because, I mean, he was already throwing the creationist paradigm out the window and saying life evolved. This was a big step. But the last thing he wanted to do was get embroiled with the church over humans. So he just literally, that last sentence says, light will be shown on the origin of humans. And it wasn't until 1872 that he actually wrote The Descent of Man and directly included humans into it. But by 1872, everywhere in the world but the United States, evolution was already, natural selection and evolution were already completely accepted. The United States being the one developed country outlier that never really fully bought into it, and still doesn't if you read the Pew and Gallup polls. 50% of Americans literally believe in a recent creation of the earth straight out of Genesis, and another 25% in intelligent design. About 25% of Americans last year, and this number is held for decades, believe in naturalistic evolution. Now, we're here in upstate New York. We probably have different opinions on average than most of the country. So you might think that's crazy, but, go to Atlanta. Yes?
I'm going to risk my question. It's two parts. What would be the attraction of a psychologist? What would be the attraction between a proto-human and our human ancestor for them to have hybridized? And more than that, if it takes more than a single encounter, they have to have been in each other's presence multiple.
Well, so I have a theory. So the average dude probably, wouldn't be interested. But again, this was tens of thousands of kilometers, tens of thousands of years. These opportunities wouldn't have been incredibly fleeting. And as any police blotter will tell you, things happen. Now, I have a personal theory. Now, I just need the archaeologists to find out how far back that goes. Because there was a very famous study carried out in Scotland, of course, in which they actually, based on the smokiness of the room, the amount of alcohol, and all sorts of other things, predicted the beer goggle effect. So it's just a theory, and this might be my fifth one to be shot down. But it wouldn't have to be often. Again, we're talking every 2 to 3 generations in an entire population, a successful mating event occur.
Which you believe was from the cell?
I mean, the Neanderthals were like twice as strong as modern humans, and we're pretty certain that there was very little Neanderthal females being impregnated, so it would have to be the other way. But the modern human population was almost certainly larger and did have a more sophisticated tool technology. So I don't want to get too deep into predicting there, but based on the evidence we have, it looks more like male introgression into the Neanderthal male introgression into the modern human gene pool, which actually goes against all historical precedent of dominant and subdominant populations. It's almost always the dominant population, males, impregnating the females of the others. So this goes literally exactly opposite of that. So we need many more Neanderthal individuals, and it'd be nice to get some now early modern individuals, and maybe we can begin to tease apart the percentages and the directionality of these gene flow. Unfortunately, for some bizarre reason, maybe the Creator is against us, the 3 Neanderthal specimens and the 1 Denisovan specimen are all female. So we don't know what their Y chromosome looks like. But if that was the case of Neanderthal males impregnating modern females, their Y chromosome should show that signal. Or the human Y chromosome should show the Neanderthal signal. We just do not know what the Neanderthal Y chromosome looks like to date. 3 years from now we might. But it's an interesting speculation. Yeah.
I have 50 questions, but I'll just ask one. Do you have a pet theory about what happened 200,000 years ago to create the bottleneck? Is it something more than just climate change?
No, is the short of it. Whatever wanderlust, it may have been technological innovation that just let that one population do it. And the reason it was a bottleneck, it was just a few people. It wasn't like a purposeful bottleneck. They may have been trying for 100,000 years before, and, like Jamestown and, other things just utterly failed and not succeeded after leaving Africa. So I'm not sure, if it's just sheer blind dumb luck, temporarily climatic events, just a couple 100-year cycles can dramatically change, the vegetation patterns in the Middle East and Arabia and stuff like that. So, like most of evolution, I'm just think it's probably blind dumb luck that one group was the one that made it. Yes?
If offered that 800 or 1,000 hybridization events would be sufficient to explain, is there evidence that limits it to that? Could it be 100,000?
Well, the same models that predict that, if there was more, we would have much larger percentage of our genome. Again, 1 to 4% or 2.5 on average it's a tiny, tiny amount. If it was at all common, it should be, 20, 30%. And so the fact that it's so small, under the best, but these models are complicated. You got to predict the demography, you got to predict the, mating, were they selected against or not. So there's a million parameters to these models. They're not trivially simple models. But, they've run many, many, many different models, and the ones that fall out under the likelihood framework are all extremely low levels of hybridization. No model accounts for a high level of hybridization. Yes? Sometimes it's who paid you. So Australopithecus boisei, now known as Paranthropus boisei, the Boyce Foundation funded Louis Leakey's fieldwork. Kenyanthropus platyops. They were sucking up to the Kenyan government because they needed new permits for their field season. There is a Monty Pythonithecus snake. Sometimes you name it after your favorite, characters or whatever. So hominins, lately the trend seems to be after the local area or local people. Australopithecus afarensis, Sahelanthropus chadiensis. Those names seem to be going, but taxonomy is great because you get to do it. I mean, you can pick any name you want. You just got to build it on the proper sort of Latin. You got to Latinize it, basically. But so yeah, you can. If you find an absolutely new species, you can name it whatever you want. Okay. Well, so we do have some limited we don't have a whole genome yet of a Native American, but from everything we know, the Native Americans are derived from East Asians 15 to 20,000 years ago. So their origins are after all of these events that we're talking about. So to study these events, they're actually not relevant, though Native Americans do have that 2.5% Neanderthal ancestry on average that you find elsewhere in Siberia and in East Asia. I do not know if they have Denisovan ancestry. That hasn't been actually explicitly looked at yet. Based on the pattern of Denisovan ancestry, I would predict they might not, because we don't find it elsewhere in Eastern Asia. We only find it in sort of central Siberia going down into Indonesia. So I would predict it's not in Native Americans, but that's what science is about. Here's a prediction. Somebody's got to start crunching the data to do it.
I was just wondering if you would want to expand a little bit on your hypotheses about yours, the red.
Oh, that's just crazy. I mean, we know that island populations of mammals typically get dwarfed. So you have pygmy hippos, pygmy rhinos, pygmy elephants on islands in Southeast Asia. It's a very common phenomenon. And it's endocast. If you actually look at the shape of the brain that's preserved in the best skull, it looks remarkably like a miniature Homo erectus brain in its shape. It doesn't look like a modern human microcephalic brain in its shape. But microcephaly is a pathological condition, and there's many different ways it can happen and all these things, so that's not a perfect comparison. But its teeth are really weird. It's just really weird. And it's extremely isolated. It's on a very isolated island. And so if any poor erectus is going to get stuck on an island, this is Gilligan's Island there. And it just happened to be for 1.8 million years, not a 3-hour tour. And they got pig meat and that's it. It's just speculation, though, because you talk to the paleontologists and they're 50-50, modern human, not modern human. Some of them are saying it's an Australopithecine. It's like crazy, but maybe. But so it's just pure speculation. And unless we get more specimens or get DNA, it's going to be hard to answer. I doubt we're going to get DNA, given the preservation conditions there, but maybe just a nice, perfectly dry burial, just maybe, but we don't even have other specimens yet. So, and so if it is a pathological modern human, we were so unlucky, the one we found is the 1 out of a million people with this pathology. What happens if we find 5 of them? Well, then, are they related? And is it just a pathological family? Well, so we need data. We always want more data. That's a good prediction. We need more data. I won't be wrong.
One more question.
Okay? Well, so for instance, the immune system. One thing I glossed over is that of the genes we got from Neanderthals and East Asians got from Denisovans, more alleles from the immune system passed through this hybrid barrier than others. So this might tell us what the ancient disease patterns were of these people, and they have, by hybridizing with us, increased the genetic diversity of our immune system, which is always helpful when fighting off things. So if there was a disease 200,000 years ago in Asia that's lying dormant since then, but it reemerges, we might have some of the alleles to fight it off left over from our deep ancestry. That'd be kind of cool, you know. Something comes out of the permafrost of Siberia. It's been frozen for 200,000 years. We warm it up, dig it up. Now we got a new disease spreading around. We might have the alleles left over from when that disease was last running around Siberia.